All right, let's just jump right in. Tucked inside nearly every single one of your cells is the complete instruction manual for you, your DNA. And every time one of those cells divides, it has to pull off this mind-boggling feat of copying that entire manual perfectly. And believe me, this isn't like hitting copy on a printer, it's a high-speed, high-stakes molecular challenge. I mean, really think about that for a second. The human genome, it's got about 3 billion base pairs. If you tried to type that out, you'd end up with a stack of books towering 200 feet high. And yet somehow, your cells manage to copy that entire library almost flawlessly every single time they divide. The question is, how on earth is that even possible? So the mission is pretty straightforward. Before any cell can split into two, it has to make a complete, perfect copy of its DNA. This is non-negotiable. It's what ensures that both new daughter cells get the full set of genetic instructions. This whole process is literally the foundation of how we grow, how we heal, really how life itself continues. The name for this process is semi-conservative replication. Now, that sounds super technical, but the idea behind it is actually beautiful and simple. Imagine you have a zipper. When you unzip it, you have two separate sides. The cell takes each of those original sides and builds a brand new matching side for each one. So every new DNA molecule is a hybrid, half old, half brand new. Okay, so that's the goal, but to even get started, the cell has to solve a major problem. That genetic code isn't just sitting there out in the open, it's locked up tight inside that famous double helix. So, step one, we've got to get inside. To crack the helix open, the cell brings in a special little enzyme called helicase. The best way to think of helicase is as a high-speed molecular unzipper. Its entire job is to latch onto the DNA and just start plowing forward, breaking apart the hydrogen bonds that hold the two strands together. And when I say high speed, I mean it. This is where it gets crazy. In bacteria, this little helicase motor can spin and unravel the DNA at an incredible rate. We're talking 10,000 RPM. It is a genuine biological machine working at an insane speed. So as helicase is tearing down the track, it opens up the DNA into this Y shape, and this structure is called the replication fork. You can think of this fork as the active construction zone. This is where all the real copying action is about to happen. So, the helix is open, the construction site is ready, now it's time for the star of the show. The cell needs to bring in its master builder, the enzyme that's actually going to assemble the new DNA strands. And here it is, DNA polymerase. This thing is an absolute marvel. It scoots along the original unzipped DNA strand, reading the sequence of letters, A, C, G, and T, and it grabs the matching partner for each one, building a brand new, perfectly complementary strand right alongside the original. But there's a catch, a big one. DNA polymerase has a very strict, very annoying rule it has to follow. It can only build in one direction. It can only add new DNA letters onto what's called the 3' end of the chain. It's like a car that can only ever drive forward, no reverse. And this one little rule creates a huge puzzle. Now, for one of the original DNA strands, this is no problem at all. We call this the leading strand. It's pointed in the right direction, so as helicase unzips the DNA, polymerase can just cruise right along behind it, building one long, continuous, perfect new strand. It's smooth sailing. But what about the other strand, the one pointing the wrong way? This is where things get really clever. This is where the cell has to pull off an incredible molecular trick to solve a puzzle baked into the very structure of DNA. And here's the heart of that puzzle. The two DNA strands are anti-parallel. They're like a highway, but the two lanes run in opposite directions. The leading strand is going the right way for our one-way builder, but the other one, the lagging strand, is going the complete opposite way. The construction zone is opening in one direction, but our builder can only work in the other. How do you solve that? This is the big question. You can't just ignore half of your DNA, right? So how does the cell manage to build this lagging strand when its master builder enzyme is fundamentally facing the wrong direction? The solution is just brilliant. The cell doesn't even try to build it all in one go. Instead, it builds the lagging strand in short, little pieces. These pieces are called Okazaki fragments, and each tiny fragment is built in the correct direction, even though the overall process is happening in reverse. It's almost like a little dance. First, a tiny little RNA primer gets laid down to mark the sorting spot. Then, DNA polymerase jumps on and quickly builds a short Okazaki fragment until it bumps into the piece it made before. Then it hops off, jumps backwards to a new primer that's just been laid down, and does it all over again. It's stitching it together backwards, piece by piece.
Okay, so we're almost there. We've got 2 new strands of DNA, but it's not quite perfect yet. The lagging strand is still in a bunch of separate pieces, and it's got all those temporary RNA primers mixed in. So it's time to call in the molecular cleanup crew to finish the job.
First up, an enzyme called exonucleoase comes in. Its job is pretty simple. It moves along the new DNA and basically acts like a pair of scissors, chopping out all of those temporary RNA primers that got everything started. But, of course, this leaves little gaps in the DNA backbone. And that's where our final hero comes in, DNA ligase. This enzyme is the molecular glue. It follows behind, sealing up all the little nicks and gaps between the Okazaki fragments. It connects everything together, creating one single, continuous, and now totally complete strand of DNA. The folks at the DNA Learning Center really put it best. They said the molecular reality is very different from the iconic image of the double helix neatly separating into 2 DNA copies. And isn't that the truth? What we just saw isn't a simple, clean split. It's a buzzing, dynamic, unbelievably complex machine at work. So why get into all these nitty-gritty details about enzymes and fragments? Well, because understanding this one single process is fundamental to understanding, well, pretty much everything about life, health, and disease. This is how you grow, how your body repairs itself after an injury. When this finely tuned machine goes wrong, when it can't stop copying, that's at the very heart of what cancer is. Our ability to solve crimes with DNA fingerprinting, that relies on these very